today's lecture is on proteomics and systems biology. The lecture outline. First, I will start on proteomics and then we will move on to the systems biology. Today, we will continue on first proteomics and then how different type of omic data can be applied for systems approach analysis, that will be discussed. Now, central dogma concept, which is an orderly and unidirectional flow of information encoded in base sequences of cells passed on from DNA to RNA and then to the proteins. This is the simplest definition of central dogma. The genome sequencing projects, they have provided researchers with an unprecedented information of genome sequences. However, there are numerous proteins which can be encoded by the genome. Therefore, analysis of the static genome by doing sequencing alone is not sufficient. A gene can code for several types of proteins because of its alternative splicing and post-transcriptional and post-translational modifications. Therefore, it suggests that studying proteins is more challenging than genome or transcriptome. Therefore, proteomics has great significance to understand the biological systems. The completion of genome sequencing projects of several organisms, including human, has been one of the most remarkable achievements of this century. However, these have not been sufficient to unravel the mystery of complex biological processes. The similar gene numbers of many diverse group of organisms has failed to explain their varying biological complexity. A more meaningful understanding of biological function can be obtained through the characterization of products of gene expression, the protein, which serve as ultimate effector molecule of biological systems. The proteomics refer to the study of entire protein complement expressed by an organism at any given time. While the genome of an organism is mostly static, the proteome is dynamic and it changes with environment and time, thereby elevating its complexity level. The gene regulation is regulated by several post-transcriptional and post-translational modifications, due to which the number of proteins expressed in a cell is much greater than its genomic counterpart. The proteomics aims to decipher the structure and function of all proteins in a given cell under specific conditions and to obtain a global view of cellular processes at the protein level. Study at the DNA level known as genomics, RNA level transcriptomics and protein level proteomics. Analysis of the proteome involves protein extraction, separation, identification and finally, characterization of various proteins. Very broadly, you can group these technologies as gel based methods, gel free mass spectrometry based methods, mass spectrometry techniques, techniques to study the protein interactions and structural proteomics. So, as we have seen, there are large number of proteomic technologies which are currently available for various applications. Many times, to address one biological question, different type of methodologies come together and then provide solutions to that problem. For example, looking at some clinical sample for identification of biomarkers of a disease, one can employ the samples such as tissue or blood or different type of body fluids, and then either directly extract the protein and subject to the mass spectrometry, or first resolve on two-dimensional electrophoresis followed by identification on mass spectrometry, or take these samples directly apply on the microarray based platforms and then detect using label based or label free methodologies. Eventually, these type of results will enhance knowledge for the monitoring the therapy response as well as identification of early disease diagnosis. This is just one example. Similarly, multiple type of proteomic technologies can be used for different applications. There are several proteomic techniques which are employed for studying these proteins, such as two-dimensional gel electrophoresis, mass spectrometry, protein microarrays, as well as some label free detection techniques such as surface plasmon resonance SPR. After discussing proteomics, now let us talk about systems biology. So, what is systems biology? Is that an historic knowledge, a method to understand biological systems, or a tool to solve the practical problems? Systems biology is the examination of a biological entity as an integrated system rather than the study of its individual characteristic reactions and components, which is termed as systems biology. A study of all the mechanisms underlying the complex biological processes in the form of integrated system of many interacting components is studied under systems biology. The systems level understanding of biological networks require information from different levels. As you can see, from DNA to RNA to proteins forming systems, and then that information can be applied to understand a complex system for different organisms. The biological information is represented by the networks of interacting elements and dynamic responses to the perturbations. These networks provide insights which cannot be analyzed from the isolated components of the system. The common elements of the systems biology include networks, modeling, computation and dynamic properties. The different type of biological networks such as protein-protein interaction networks, gene regulatory networks, protein and DNA interaction networks, protein, lipid, protein other biomolecules network and metabolic networks. The various ingredients of systems biology. For example, if you are studying about a cell and the system's behavior, one need to look at the genome, its transcriptome profile, proteome profile, how protein, DNA and different type of transcriptional networks are altered, protein-protein signaling networks, multimeric complexes, how they are formed, how protein is localized in the intracellular dynamics and metabolic networks. So, all of these are ingredients of studying about a system. Now, systems biology study can be done at different levels. For example, to study the complex physiology of human, one can look at individual systems such as respiratory systems, nervous system or other physiological systems. studies can be done at the intercellular or intracellular level, and finally at the molecular level involving genomics, transcriptomics and proteomics. So, why there is need for systems biology? The study of biology at the system and subsystem level for understanding the biological processes and network is very much required. As you can see, to understand even simplest system of a cell, how it is regulated with its extracellular space, the cytoplasm and different other components, examination of the structure and dynamics of cellular and organismal function is very much required for understanding the systems, rather than the characteristics of isolated parts of the cell or organism. So, what is the aim of systems biology? To understand the biology in holistic approach rather than the reductionist approach. The systems biology aims to quantitate the qualitative biological data and provide some level of predictions by applying different type of computational modeling. The systems biology approach involves first of all collection of large experimental data sets and then mathematical models to provide insight of some significant aspects of the data sets. A simple systems biology approach would involve experiments by adding new data sets, which will be used for model constructions and model analysis, and the biological insight derived from these models can be used to propose new hypotheses. So, the properties of a systems are probably more than just the sum of all its individual properties or its components. Therefore, it is possible that system may have its own property by applying all the components. So, what are different approaches have been taken to study the systems biology? The distinct approaches of systems biology include the model based and data based methods. The model based approach involves some prior information which can be implemented in the models, whereas in the database objective is to find the new phenomenon. The model based relies on computational modeling and simulation tools, whereas the database method relies on the omics data sets. So, in model based systems biology approach, it is difficult to build the detailed kinetic models, but in database system, the complex relationship among various type of omics information and metabolic pathways and networks can be created. Studying systems component is very challenging. Systems biology and biological network modeling aims to understand the system's structure and function for better understanding of system properties like its robustness, as well as used for the prediction of systems behavior in response to the perturbations. The reductionist approach involves disintegrating the system into its component parts and studying them, whereas the integrative approach involves integrating the study of individual components to form conclusions about the system. What is systems biology triangle? So, first of all, the system's information is generated at various levels. As we have discussed, starting from genes to mRNA to proteins to metabolite, or identifying regulatory motifs, metabolic pathways, functional modules and different large scale organizations. This information has to be stored, processed and further executed to identify the system level information. Even simpler systems such as cell can be linked with various properties. Its genome sequences of different molecules, intracellular signals, transcription factors, different type of cis binding activities, the expression profiling of RNA and proteins, and different type of cellular processes. So, what approach one can take to study about the system? Extraction and mining of complex and quantitative biological data, integration and analysis of these data sets for the development of mechanistic, mathematical and computational models, validation of these models by retesting and refining after proposing some hypothesis. Different online databases and repositories are nowadays developed for sharing large data sets and various systems models. The systematic approach to study how molecules act together within the network of interaction that make up life is definitely going to be useful to understand the systems biology. The systems biology triangle, as you can see here, involves the experimental data set could be derived from different type of omics platforms, technologies, how the computational analysis can be performed, different type of bioinformatics softwares and tools, and then computational modelings by obtaining some theoretical concepts. The synergistic application of the experiment, theory and technology with modeling to enhance the understanding of biological processes as a whole system rather than the isolated part is termed as systems biology triangle. So, the systems biology triangle, the wet lab experiments or bioinformatics based data analysis can be used to propose a model. The model building as an aid to understand the complex system, and some hypothesis can be generated which could be used further to propose more quantitative models or predictive models, and also it can be used for independent techniques for the model validation. So, what is system study? First of all, the difference between systems study and the component study one need to understand, and this is what we have tried to emphasize in the previous slides. After generating the data set and creating the systems biology triangle, then this information can be used for understanding the systems in the more complex and mechanistic level. Systems study and model building. The system science includes synthesis, modeling concepts, analysis. Life sciences discipline provides quantitative measurements, genetic modifications and deriving some hypothesis. The information sciences enables the visualization, the modeling tools and different databases. So, this model building as an aid to understand the complex system is very useful for systems level investigation. System is an entity which maintains its existence through a mutual interaction of its constituent parts. The systems biology research consists of identification of the parts, characterization of the components, exclude the ones which are not a part of the system, identify the interaction of the components with each other and identify the interaction of the components with the environment which modulate the parts either directly or indirectly through modulation of internal interactions. The systems biology concept can be understood with help of two approaches such as reductionist approach and integrative approach. The reductionist approach focuses on disintegrating the system into its component parts and studying them, whereas the integrative approach focuses on integrating the study of individual component to form conclusions about the system. Consider a cell with its component molecules. Let us say we want to study the metabolic pathway as a biological system. When the environment of the cell is perturbed a little, the individual components undergo unique changes such as increase in the production rate or decrease in their amount. At this stage, due to lack of knowledge of the nature of interaction of proteins, we cannot interpret how the system gets affected. But when we study the interaction of one component with the other, we can conclude that the increase in rate if DNA binding protein leads to increase in the synthesized amount of DNA, which further changes the final amount of lipoproteins produced. Thus, we can see that to study a system, we need to analyze not just the components, but their interactions. These biological systems can be protein-protein interaction networks, gene regulatory networks, protein DNA networks, protein lipid networks, and metabolic networks. To study the system, we need to know about the components and its interactions. The data about the components comes from genomic and proteomic studies. The information about the molecular interactions comes from the interactomic studies. Here it is shown that in a systems approach, experiment, technology and computational modeling, this triangle is very important, which has to be linked with the theory to form a systems triangle. Let us now talk about how to model the biological networks. To build a model in systems biology, first of all, the parts list can be generated by using data sets derived from the systems biology approaches. The systems or subsystems model can be generated, which can be used for the systems model analysis. Now, this could be applied for the real systems, and by applying the knowledge using bioinformatics tools, it could be again applied back to the original components, which could be used to derive some hypothesis and validation of these data sets. So, it will work like a closed loop. To build the models in systems biology, information is generated at different levels. Level 1 such as DNA and gene expression, level 2 the intracellular networks, level 3 cell and transmembrane signals, and level 4 integrated organ level information. What are the frameworks required for the modeling schemes? Different type of deterministic or stochastic models have been proposed. The compartmental variables or individual or functional variables have been studied. The spatially homogeneous or spatially explicit models are generated, which could be applied in the uniform time scale or separated time scales. Now, this framework could involve single scale entities or cross scale entities. As you can see here, this framework requires different level of information in very complex manner, whether it is curation of the databases, how to align these information using bioinformatic tools to generate the predictive models, which could be also developed by using the literature curated data sets or experimental data sets, and finally, it could be used to study the systems level properties. Let us discuss the workflow of mathematical modeling. A paradigm can be proposed based on modify, model, measure and mine. So, systematic experiments, different type of molecular genetics, chemical genetics and cell engineering approaches can be used for modifying, and different level of measurements by applying microarrays, spectroscopy imaging and microfluidics based approaches from proteomics and genomics can be used further for mining, which involves bioinformatics, databases and data semantics. Now, these data sets could be used to derive the models, which could be reaction, mechanistic, statistical or stochastic models. So, starting from systematic experiments to reaching and deriving the quantitative models, this workflow can be applied. The modeling of probabilistic processes involves, let us say you want to study a biological system. So, some experiments has to be performed, the experimental data sets will be generated, from which some statistics can be applied, which can be used for the comparison. Now, different type of models can be generated by using simulations and simulation data sets, which can be used for intermediate statistics, and by comparing these two type of information and adjusting the parameters, one can study the systems and derive the probabilistic processes. So, what is ordinary differential equations and stoichiometric models? The quantitative analysis measures and aims to make models for precise kinetic parameters of a system's network component. It also uses the properties of network connectivity. The ODE is a mathematical relation that can be used for modeling the biological systems. The quantitative models mostly use ordinary differential equations or ODE to link the reactants and products concentration through the reactions, reaction rate constants. To develop the computationally efficient and reliable models of the underlying gene regulatory networks, these ODE models can be used. The stoichiometric model, it is modeling a biological network based on stoichiometric coefficients, reaction rates and metabolite concentrations.